Today, we are going to go over the process of cell division, including the phases of mitosis. But first, let's talk about why cells need to divide. So, number one, they need to make sure they are staying small enough so that they can efficiently work. So, what this means is if they get too big, they're going to have problems with getting in enough needed materials and also getting rid of all of the waste products that they will be creating. Number two, reproduction. So cells that reproduce asexually, they are making clones of themselves. So all the offspring will be exactly the same and the same as the parent cell. And there's two types of this. So we have binary fission and budding. So binary fission, this is a process that a bacteria cell would use. So you start with one cell and you can see it just splitting into two. And now the two daughter cells have the exact same DNA as each other and as the parent cell. Budding is a process done by an organism called Hydra, and it is just having this appendage come off, and that new appendage is just a separate organism now, and it is exactly the same as the original parent Hydra. Another reason why cells divide is for growth and to repair and replace cells. So, if you get a paper cut, for example, you have to regrow skin cells. Or this puppy is going to grow into an adult dog, and to do that, he'll get bigger and need new cells. Another fact is that your body is making red blood cells all of the time. It says about 6 million red blood cells every few seconds. And so, whether you're making new red blood cells or if this puppy is growing and turning into a dog, if you are making new skin cells to heal a cut, you are using the process of mitosis to make these new cells. And so, mitosis is just a very small part of the cell cycle. The cell cycle is made up of three parts. You have interphase, mitosis, and cytokinesis. Mitosis is the part where the nucleus and the DNA in there is actually dividing and getting split up. Cytokinesis is the part of the cell cycle where the cytoplasm and all of the other organelles are getting divided into two cells. And then interphase is making up the biggest portion of the cell cycle. And what do you think has to happen here before a cell can actually divide? Well, a cell has to grow and get ready to divide and make a copy of the DNA before it can split into two. And this interphase section is actually divided into three parts: G1, S, and G2. The G is going to stand for growth. So during this portion of the cell cycle, a cell's just growing and getting ready for division. The S stands for synthesis. This is where a cell is going to make a copy of all of its DNA. And then we have G2, and that is just a little bit more growth, and the cell is getting ready to enter mitosis. So interphase, again, is made up of three parts. We have G1, S, and G2. And once a cell goes through those three parts of interphase, it's now ready to go into the division section of the cell cycle. And so the M phase is made up of mitosis, which is the division of the nucleus, and then cytokinesis, which is the division of the cytoplasm. When a cell is in interphase, it's important to know which type of DNA it's going to have here. So which form of DNA? It could be chromatin, which is the long and stringy form of DNA, or it could be that chromosome type, where it's all tightly coiled up into that X shape that you know. But what the correct answer is chromatin. So DNA has to be in that long stringy form so that the proteins can get in there and read it and make a copy of it. And this is also a reason why cells are mostly in interphase. And it's because a cell that is actually doing its job and just making the proteins that the body needs, it's going to be in this stage where the DNA is in the long stringy form so that it can be read and used actively to make all the proteins that a cell needs. So a cell is mostly in interphase, in G1 specifically. That's where it will spend most of its time. And then when a cell is ready to divide, the chromatin will then condense into a chromosome. And remember that a chromatid is a half of a chromosome, and it's going to be connected by something called a centromere. And now once our DNA is all bound up in that X-shaped chromosome, we are ready to enter mitosis, which is made up of these 4 steps. We have prophase, metaphase, and anaphase. Or prophase, metaphase, anaphase, and telophase. We forgot telophase. Don't forget that. And so prophase, you remember, it stands for prepare. This is when chromatin condenses into chromosomes, the centrioles appear, the spindle fibers form and attach to centromeres, and the nuclear envelope is actually going to go away. Metaphase stands for middle. This is when the chromosomes line up in the middle of the cell in a nice, even row. Anaphase stands for apart. This is when the spindle fibers pull the chromatids apart to opposite ends of the cell. And then for telophase, that's where our cell is splitting up into 2 cells now. And then towards the end of this stage, that's when the nucleus is going to reform around each of those sets of genetically identical DNA. And then after mitosis, you have cytokinesis, and that's just where the cell is officially and finally splitting into 2 completely different cells. They do have the same exact DNA, though. And you need to know that in plant cells, this is a little different because they have something called a cell plate, and that just helps them divide a little bit more easily. And they have this because they have a cell wall, so plant cells are a little different from our cells. So a summary of the cell cycle here. We have interphase, which is made up of G1, S, and G2. We have prophase, then metaphase, anaphase, telophase, and then lastly, we have cytokinesis, which just leads to the creation of our 2 cells. So we're just going from 1 cell to 2 cells. That's the whole goal here, is to make new cells that are exactly the same. And you do this whenever you're growing or you need to replace a cell. And it's just important to know that the 2 cells that get made are exactly the same as one another. So in this way, meiosis is very different from meiosis. And we will learn and go over more about how these 2 processes are different in the future.